So we'll get back to the lecture. So we'll now move on to talking about the motor unit and then to muscle contraction. So we talked briefly previously on the alpha motor neurons and their role in this the motor pathway, a somatic pathway, the voluntary that causes muscle contraction. So one alpha motor neuron will innervate many muscle fibers, and that, as we talked about, is termed the motor unit. So the actual potential will come down from the higher brain centers and go down the length of the nerve, and it will, you can see the myelin sheaths here, the nodes around the A. We'll talk about it again a little further, and this is the direction of propagation of the action potential as it jumps from node to node, these myelin sheaths. And then as the action potential gets to the neuromuscular junction, what you're seeing here is the nerve and all of the fibers that it innervates. This is known as the motor unit. And when this contracts, all of these fibers contract. And that is the all or none principle that somebody mentioned earlier. We'll talk about again. So when this contracts, all of these fibers will contract. Now, fibers right next to it may remain sedentary. That's about how much force is necessary. Well, once it's activated, all the fibers that are innervated by that nerve will be activated and contract. That nerve and those muscle fibers are termed the motor unit. Okay? So the יחידת מוטורית, העצב וצירים של the innervation. And this is an electron microscope of the motor unit. As you can see here, this is the motor end plate, neuromuscular junction, the muscle fibers, and the alpha motor neuron. So once this is activated, the acetylcholine will be released from underneath this neuromuscular junction. It allows the action potential to proceed down the transverse tubules, cause contracture of the muscle, of the fibers of that particular that's innervated by that particular neuron. Now, as I mentioned to you, that only the fibers that are innervated will contract. And here we can see is two different motor units. One motor unit is active, the other motor unit is not. And only the motor units that are active will actually contract, while the others that may be adjacent, right next to it, צמוד לצירים האחרים, אם אין להם גירוי, לא יהיה קיבוץ. They will not be recruited.
Professor, when does that happen, that very close motor units?
It's happening right now to you. As you pressed on something to speak to me, you didn't need many motor units to activate that. So some motor units in your hand were activated and others were not.
But they're that big, the motor units?
No, they're not that big, but the fibers, again, is the smallest. Keep in mind that we talked about the muscle, right? It's divided into fasciculi. In the fasciculi, you have the myofibril. You have the fibers. They have the myofibrils, right?
Yeah.
So you go down to it's the smallest functional unit, the fibers, or the sarcomere in the fibers. So you don't need your entire muscle to contract. You only need to have several fibers in the muscle to contract. The motor unit is only those fibers that is innervated by a specific nerve. So take a look at the picture of it a little better. You have a decision being made. You have a sensory organ that says I need to move something, and it goes up through the dorsal pathway, right? Sensory pathway. Message is sent down the motor unit through the ventral horn to create a response, an effect. That response, as it gets to the muscle, is through nerves. Nerves coming from the spinal column, then bifurcates into various nerve fibers that goes into the motor unit. And at the motor unit, you have it's at the neuromuscular junction that it innervates, as you can see a bigger picture of that here, of various fibers. that as the acetylcholine is being released as a neurotransmitter, it will cause an action potential, and only those fibers that are innervated will actually contract. The other fibers that are not innervated will not contract. So this way you have gradations of force capability. So you're not all of a sudden lifting maximally on everything that you're trying to do. If you're trying to lift your wallet, לא צריך הרבה כוח. אבל אתם רוצים להורים 100 קילו, אתה צריך המון כוח. אתה יכול לגייס כל היחידות מוטורית. אבל אנחנו לא צריך, אולי 2% של היחידות מוטורית ביד, את צריכה לעלות את זה. You have this ability gradations in strength.
מי מחליט כמה יחידות מוטוריות ישתמשו? יופעלו? איך, אבל בתת מודע או דרך חוט השדרה?
Listen, if you want to give a maximal effort to lift up your wallet, you can do that. It may not be the smartest thing you've done in your life, but you can do that. And if you didn't want to activate a lot of motor units to lift up a 100-kilogram bar, you can do that as well. But if you really wanted to lift the bar, 100 kilograms, you're going to have to recruit enough of the motor units that will allow you to be able to successfully lift that bar. So it's not pressing a button and say, okay, it's not like a computer where you set something to lift it. It's a desire that you're exerting more force, and the more force that you desire, it's going to create a greater activation of the motor unit. So it's electrical signals. It's not a pre-programmed response. You're not a robot. You still have to think about it. It's a desire. Okay. So let's talk now about muscle contraction. Do this several times slowly so you understand what's going on. So what you're looking at here is the sarcomere. You're looking at the two myofilaments, myosin, the thick filament, and actin, the thin filament. And by this point, you should be able to recognize what you're looking at here. The myosin thick filament has two globular heads. The actin has three proteins. You have the actin molecule, you have the tropomyosin, and various parts you have the troponin. Now, what's important to keep in mind is on top of the troponin molecule, you have what is known as the calcium binding site. And that's found here on the troponin molecule. So calcium comes, and you have the electrical signal, right? Now calcium is released from where? Where's calcium released from? Plasmic reticulum, correct. So it's released, you got the electric potential went down the transverse tubule, activated the pool of calcium in the sarcoplasmic reticulum, released it, and then goes to the troponin molecule. And what calcium's role is to cause a conformational change. It's going to cause a movement of troponin, forcing a movement of the tropomyosin molecule. And as it moves, it's going to expose the actin binding site. The myosin head then attaches. So calcium comes, attaches to the troponin molecule. It causes tropomyosin to move. As it moves, it's going to expose the actin binding site, and myosin is going to attach to the actin molecule, and it's going to cause a ratchet type of movement that creates muscle contraction.
So it's like a ligand gate?
I'm sorry?
It's like a ligand gate?
I wouldn't call it ligand gate, no. It's causing this, it's a filaments to slide over one another. So what's happening is the myosin head attaches, it causes a ratchet, and this type of movement of as it ratchets itself from one side to the other, it creates that movement, shortening the H zone. Now, here's the key. We've talked about ATP. Everybody knows what ATP is, right? What is ATP? ATP is the fuel of the... Now, where does ATP become important? It's right here. Once calcium came, it created the opening of the active site. Myosin attached. What creates the dissociation This is an important word. The disassociation from the actin to myosin, so you continue the contraction. וההפרדה בין actin ומyosin. It's ATP. This is what fuels this ability to separate. And on the myosin head, you have an actin binding site. ATP comes to the actin head. As it attaches, it releases. And then the contraction can continue. And that's how we have the shortening. As the actin and myosin filaments slide over one another, shortening the H zone. So with calcium has its role of opening up the active site, causing a conformational change with tropomyosin and troponin, opening the active site on the actin molecule. The myosin head attaches to that active site, and then it's released by ATP. And then the whole process begins again. Is there any time in life that we ever run out of ATP? Never. In fact, we run out of the ability, the rate at which to supply it, right? We're doing high intensity exercise. We can't produce ATP very quickly. So we get into fatigue. Our ability to disassociate becomes reduced, and we have to slow down. In endurance activity, we have plenty of ATP being produced, plenty of energy to produce ATP, and we can go on theoretically for as long as we can. And that's the difference between aerobic and anaerobic activity, high intensity and lower intensity activity. The only time that we run out of ATP, is no longer being produced, is at death. Manshal Mavet, ATP. And what happens at death? If the muscle is contracted, you have a situation known as rigor mortis. And the muscle gets locked into the position it was when the person passed away. So you see someone who passed away a while ago wasn't treated right away. The person was by themselves, and they've been in an awkward position, and they're very stiff as you get to the body. That's rigor mortis setting in. And they ran out of ATP, and you couldn't separate the limbs. That's an example of when no longer ATP, it's called rigor mortis.
What's the difference between the high intensity and low intensity?
Well, the ability to supply ATP. So you wouldn't, if you're going high intensity activity, you would fatigue quicker because you wouldn't be able to produce ATP fast enough, so the contraction would have to slow down. During low intensity, you're producing ATP at sufficient rate that you can continue exercise till other factors may make you fatigue.
Okay, I have a question. When it contracts the myofibril, does the actin binding site, like the myosin head, it jumps from one troponin to another?
Correct. So it's causing the sliding film moves from one part of the actin, the other part, and it keeps on sliding across. And that's how we have contraction from the current. We have contraction.
Can you explain again what happens after ATP attaches to the actin binding site?
No, ATP does not bind to the actin binding site. ATP binds the myosin head. So what happens, ATP binds the myosin head.
ה-ATP זה סוג של כאילו תעלת קישור, כביכול?
ATP הוא chemicals, adenosine triphosphate.
סליחה, לא הבנתי?
ATP is adenosine.
כן, לא, I understand that it's something that regarding to energy, but I don't understand איך זה כאילו עובר מהרשת הירוקה לראש של המיוזין.
It's being produced in the muscle. Where's ATP produced?
Mitochondria.
Mitochondria, correct. So then ATP goes, attaches to the myosin head. This is what releases the myosin head from the actin binding site. There's an enzyme on the myosin head known as myosin ATPase. This enzyme releases a phosphagen group from the ATP. From ATP to ADP, the adenosine diphosphate. You have the release of the myosin head from the actin molecule. Then the process continues until your desired effect is reached. You have the contraction. So there's constant exchange of ATP to ADP, and then it rephosphorylates it in the muscle through the process of whether it be glycolysis or oxidative phosphorylation, depending upon the type of activity that you're doing. Any questions regarding that? This enzyme, know where this is occurring, understand what is happening here.
I didn't understand.
Need to understand.
The ATP separates them?
Correct.
So what causes the movement?
The ATP is constantly being supplied to the muscle for the contraction. So calcium is being released.
Yeah, but I'm asking what causes the contraction? After the ATP separated the myosin from the actin,
The contraction is caused, the calcium, the whole process is repeated upon itself. So calcium opens up the active site, right? The myosin head attaches. ATP releases it. Calcium is released again. Contraction, still getting the stimulus, the gerui, mshich kibutz. So the calcium is still being released. So the contraction goes from one active site, next active site, next active site. You'll get complete the contraction that you want. Whether it be full range of motion or partial range of motion of the muscle, that's up to what you're doing.
So it's the ATP and calcium together, they cause the contraction?
Yes. So as long as you're still releasing calcium, which is, again, it's not a problem, it actually will then continue with the contraction. And that's based upon the stimulus from the brain. And this is again another picture of the sliding filament theory. During contraction, the H zone contracts. You ever sees this? It's important to take, as you contract, the H zone contracts in size. So this is there, it's going over each other, shortening. The H zone and I band both shorten. The maximal contraction.
Professor,
Yes.
the calcium, it's being produced by the what's it called, sarcoplasmic reticulum.
It's stored in the sarcoplasm.
But besides nutrition, does our body produce it naturally?
It's generally able to be brought up within the diet is sufficient.
Okay.
Now, muscle contraction occurs as a result of the stimulus by the central nervous system. And we talked about how that stimulus is propagated as electrical signal the long length of a nerve cell. It goes from the brain to the spinal cord to the peripheral nervous system and finally to the motor unit and neuromuscular junction. We talked as well previously on the nerves, the myelin fibers. The myelin fiber is interspace, has a specific type of cells called myelin sheaths. And these myelin sheaths have Schwann cells that surround it. And the purpose behind this is to allow for saltatory conduction. Saltatory conduction is very important for enhancing the speed of the contraction. And why it happens is because of these nodes around VA. So as you can see, here's a node around VA between each myelin sheath. And what happens is the impulse jumps from node around VA, node around VA, the length of the curve. We'll finally get to neuromuscular junction, which the action potential causes a release of the acetylcholine, which is a neurotransmitter that actually causes the stimulus to the motor unit. Okay. Nerve fibers that do not have this myelin sheath are known as non-medullary nerve fibers, and their conduction is much slower. Okay. That's important things that you need to know.
Where do we have non-medullary nerves?
I'm sorry?
Where do we have non-medullary nerves?
Probably I believe the GI tract. There's non-medullary nerve there. Also some other areas. But let me take a look at that for you, okay? That's a good question. I'll get back to you.
Thank you.
Okay, we talked about the importance of an action potential, and the nerve impulses, or as we talked about, exist in electrical energy. And what happens is these gated channels along the nerve, the electrical impulse is going to come. It's going to cause where a higher concentration of sodium on the outside will cause it to go inside the cell at the expense of potassium, which has a net negative charge in terms of the outside compared to the inside. As sodium comes in, it's generally a 2-for-1 trade. It will then become more positive and creating a threshold effect that creates the action potential. As an increase in sodium goes in, potassium leaves. It's actually not a negative, it's a positive, but just more sodium goes in and potassium leaving, creating an area of high concentration going inside to an area of low concentration. Once you get that threshold reached, then the action potential then happens. So you build up the electrical signal, enough positive charge enters the cell, and you have an action potential that gets propagated along the length of the nerve until it reaches the muscle. So let's talk about the sequence of events that leads to muscle contraction. The action potential goes from moving from the length of the nervous system, from the central nervous system to the peripheral nervous system. It gets now to the neuromuscular junction. The nerve here, and this is the presynaptic cleft, the presynaptic membrane, the postsynaptic membrane here, we have now the post. If the action potential is sufficient, it's going to cause a release in these vesicles here, these vesicles of acetylcholine. If the action potential reaches threshold level, it then releases the acetylcholine across the synaptic cleft. They go in this direction, they interact with the postsynaptic receptors, and this will cause an action potential to occur. As the action potential occurs, the electrical signal goes down the transverse tubule right, on the muscle fiber that's connected to the sarcoplasmic reticulum, releasing calcium that and then interacts with the troponin molecule on the actin myofilament, resulting in muscle contraction. So again, as the nerve cell carries the message that we want contraction, maximum potential is reached, the vesicles that contain the neurotransmitter move towards the edges here of the presynaptic membrane. They're released through the process of exocytosis that we talked about. The acetylcholine then crosses the cleft, attaches to the post-membrane receptor, creating an action potential, sending that electrical signal down the transverse tubule, releasing calcium from the sarcoplasmic reticulum. Calcium then binds to the troponin molecule, causing tropomyosin to move, to shift, exposing the active site on actin, allowing the myosin head to attach. You all understand that?
What's ACH receptor?
Acetylcholine.
What does it do?
It's a neurotransmitter.
It's specifically a neurotransmitter for muscles?
Yeah, for the neuromuscular junction.
Can I ask a question?
Okay.
I wanted to know. I didn't understand the part where how the calcium makes the myosin head connect to the actin.
Sure.
I don't. How does that happen?
Okay, so you have the actin thin filament, okay? That's the tropomyosin. Calcium is released, attaches to the troponin. You with me right now?
Yeah, that part I understood. It's
Okay.
from now. I didn't get
Okay.
it.
So once attached to the troponin, it causes the tropomyosin here to move. Simple, it causes a conformational change. תנועה של tropomyosin. ברגע tropomyosin זז, מקום פעילה, the active site, so actin is now exposed. There's a movement, expose the active site on the actin molecule, and the myosin heads of the myosin now can attach. So the role of calcium is simply to cause a change, exposing the active site on actin. That's its role. Okay?
Okay, thank you.
My pleasure. Okay, so we take another picture.
What occurs in the T-tubule?
I'm sorry? What was the question?
שומעים אותי?
I do. אני שומע.
Can you back for the slide?
This slide's going to give me the same thing, but what's your question? You need the other slide?
The previous one, yeah.
Okay.
מה עובר ב-T-tubule?
The electrical signal. The signal חשמלי.
Okay.
Reaction potential. Okay, so here, this is just above that T-tubule. What this is showing us simply is what's happening at the presynaptic membrane postsynaptic membrane. This is the fiber. This is at rest. This is activated. And what you're looking at here is simply acetylcholine, the neurotransmitter, that's being released. That's going to cause the action potential. This is in another picture, and then there's going to be a number of different slides that are going to show the same thing, just from different sources. So the first thing that's happening is we're at the motor unit. This is the neuromuscular junction. The action potential occurs, causing release of acetylcholine. As acetylcholine crosses into the postsynaptic membrane on the neuromuscular junction, it's going to cause an action potential. What it's really going to do is cause a exchange of sodium for potassium. You can see that right here. So you see the exchange of sodium for potassium. And once enough sodium comes inside the cell, and this is due to the acetylcholine, right? As you increase sodium in the cell, we get the action potential. That action potential that goes right down the transverse tubule is what this is. And the transverse tubule, what does it do? It releases calcium from the sarcoplasmic reticulum. Calcium is released, right? Leaves the sarcoplasmic reticulum, attaches to the troponin molecule, causes change in the tropomyosin, exposing the active site on the actin. You can see these active sites. Covered by tropomyosin, troponin is moved, it changes movement of tropomyosin, and the myosin head attaches. And this is what's known as after sarcomere. And you have sarcomere after sarcomere, right? Everybody see that? Guys, if you don't understand, please let me know. This is really critical in terms of how muscle contracts.
When the muscle is inactive, is the actin and myosin connected or unconnected?
It's not connected, right? If it's at rest, it's not connected.
Okay.
But then if it's not connected, how is the myosin head not exposed anyways at rest?
It's just it's in its normal place of rest. It's not, but it's not interacting with the actin. It's not causing contraction.
So when does it connect? Because the release of the calcium, it exposes it.
Yeah, but remember, it's voluntary, right? Skeletal muscle is voluntary. You control it.
No, I don't understand. At what point does the myosin connect to the actin?
When it's stimulated by the electrical signal from the brain.
When the calcium is released?
Is this Kayla?
Yeah.
Kayla, put on your picture for a second. Can you?
Hi.
Hi there. Okay. Can you have somebody? Where do you have on your left hand side? You're sitting in a nice chair. What's on your left hand side?
There's a.
Look down. Look down. Look down. What's on the left hand side? Is somebody you have at the table?
Sorry, my dog is barking. Well, on my left hand side, I have
How big's your dog? How big's your dog?
Pretty big.
Pretty big? Okay, don't lift the dog up then. What do you have small?
My phone.
Okay, excellent. I want you to lift your phone up over your head. How do you think you did that?
The action potential.
Well, you had a signal. You had a voluntary decision. I asked you to lift your phone over your head. Now, you had to activate some fibers. The phone's very light, no doubt about it. If I ask you now pick up your dog and lift him over your head, you probably have to use your legs. You have to do both arms. You'd have to now recruit additional muscle mass in order to do that. It would require more of a concerted effort, and it's more of a structural effort to do that. But you can do it. I'm sure you've lifted your dog over your head before, right?
Yes, sir.
So in order for you to do that, you have to now recruit additional muscle fibers. So what happens is you have to recruit additional motor units. So the signal from your brain goes down to the muscle that you're going to use. Now, as somebody who's lifted your phone, I'm sure about 1,000 times a day, you are very used to that sort of movement, very efficient at that movement. I asked you to lift your dog, which you probably lifted when he was a puppy. You haven't lifted in a while, maybe you have. It's a different movement. And so you're going to be recruiting those muscle fibers, motor units, in order to recruit additional motor units to allow you additional amount of force to lift the dog over your head. As you do a new move, I ask you now to lift your dog as you do a lunge. I want you to take your stand up, take your right leg, step forward, and at the same time, I want you to lift your dog over your head. You think you can do it?
No.
No. I bet you could, but would have to train you. You definitely can get to that point. So what happens?
The test, the cow.
As you begin to contract, learning how to contract, the nerves, this is what's known as motor development. You begin to train the nerves how to respond more efficiently. Muscle begins to adapt as you begin to lift heavier weight in new positions and new types of movement. So it's all voluntary. So when you lifted your phone, this is something you do all the time. Your body's used to it very light. As I said, let's take a larger object like your dog. Now you have to get up, use your legs, use your arms. More motor units need to be contracted. So that's the example how voluntarily you're thinking about, okay, I need to contract more muscle. The brain says, okay, we're going to recruit 40%, 50%, 80% of your motor units of that muscles, allowing you to accomplish that, depending on how strong you are. So if someone else in your class is twice your size, they may need not to recruit as many motor units as you need to in order to accomplish that. You understand it better now, or I confused you more?
Yeah. No, I understand.
Okay, good. Okay, nice question, though. Okay.
Professor,
Yes.
just to make sure I understood. So the calcium connects to the troponin, causes the tropomyosin to move. Then the ATP allows the myosin to unconnect from the binding site. And then because the troposin is moving, that causes the contraction?
Yeah, Aviad, you got it right. 100% correct.
Okay, so what exactly causes the troponin is what they're connected to the calcium. So how come the tropomyosin is what moves?
No, the calcium connects to the troponin, and that connection causes the tropomyosin to move. It creates a connection that allows the tropomyosin to actually change its position.
Do we need to know how exactly, or we'll get into that?
No, we really don't get into it. All we know is we're going to talk about is how the calcium attaches to troponin. That connection between calcium and troponin causes tropomyosin to expose the active site.
Okay, thanks.
So somebody brought up earlier the issue behind the all-or-none principle. And very simply, what the all-or-none principle is stating is that when the nerve of the motor unit is activated, the contraction of all the fibers that it innervates will contract. All the fibers that nerve innervates will contract. Gradations in force, and this is important for you to understand, so put a little star next to it, and I guarantee you to see a question on this, is increases in force are going to be accomplished by the number of motor units that are recruited. The increase in force, recruit more motor units.
אתה יכול להסביר את המשפט האחרון שאמרת בעברית? לא קלטתי כל כך.
all in none, אני לא חושב שיש ביטוי בעברית בשביל זה. כל כלום.
כל או לא כלום.
אבל מה קורה, הגרדות של כוח, אתה צריך לגייס יותר יחידות מוטורית. יחידה מוטורית הוא העצב וכל הציבים שעצב להיכנס. ברגע שהעצב הוא פעיל, כל הציבים ששייך לעצב ספציפי הוא פעיל. אתה רוצה לגייס עוד כוח, אתה צריך לגייס עוד יחידות מוטורית. במאמץ מקסימלי, אתה חייב לגייס כל יחידות מוטורית בתוך השריר. מאמץ לא מקסימלי, אתה יכול לגייס אחוזים בתוך זה.
So each nerve that, let's say, connected to the biceps, for example, connects to like few muscle fibers. So if I want to contract, to lift a heavier weight, I need to contract more fibers of the biceps.
That's correct.
All right.
And talking in exercise physiology, we actually talk about recruiting different types of motor units. And we'll talk about that actually today. There are two types of motor units. There's fast twitch and slow twitch. And depending on the intensity, which motor unit you'll actually recruit. There's some really important place behind that. So this is just a review of the of muscle contraction. As I said, we're going to go over this quite often. So we have the motor action potential propagated. Okay, so you have the neuromuscular junction, the action potential, the acetylcholine crosses over into the postsynaptic membrane. As the acetylcholine comes over, it's going to cause a action potential where sodium ions go into the cell, potassium leaves, creates a change in the electrical structure. Once the threshold level is reached within the cell, creates the action potential that goes down the transverse tubule, then releases calcium from the sarcoplasmic reticulum. So as the calcium is released from the sarcoplasmic reticulum, then binds to the troponin molecule that removes the blocking action of tropomyosin, exposing the active site, which the myosin head can now attach. Then it causes a movement, ratchet-like movement, between the actin and myosin molecule. ATP comes, connects to the myosin head. It's phosphagen is released through myosin ATPase. Calcium then is taken back up by the sarcoplasmic reticulum as ATP attaches. Calcium removal from troponin restores tropomyosin blocking action, and then we begin the process all over again. So this is just the sequence of events. So there's a couple of things you may want to write down what this means. AP is the initiation of action potential from the central nervous system, the release of acetylcholine in the neuromuscular junction. The acetylcholine binds the postsynaptic receptor. It opens up ionic gates, so sodium moves inside of cell as potassium exits. Once it reaches its threshold level, the action potential will occur. That action potential is propagated from the muscle surface, or the fiber surface, into the muscle fiber along the transverse tubule, where calcium then will be released from the sarcoplasmic reticulum and binds to troponin. Is everything that we're just going through a review. This is nothing new by now. You've heard of this several times today. Tropomyosin is then moved, exposing the active site. The myosin head then binds to the active site on the actin filament. This bonding will trigger release of energy that's stored in myosin that produces an angular movement of the cross bridge. The ATP then is going to bind to myosin, breaking the linkage, causing dissociation, and the ATP is split with the enzyme myosin ATPase, transferring energy to the cross bridge and getting it ready for another cycle. This 11-step process, if you know this and you can recite it by heart, you understand muscle contraction.
Professor, is it that every time I contract the muscle, this process happens?
Yes, it does.
Like if I'm doing hammer curls every single time this happens?
You do hammer curls, preacher curls, standing barbell curls, bicep curls, dumbbell curls, reverse curls. This is the process that happens all the time.
Professor, can you explain again 11 about the ATP splitting?
Yeah, so the ATP attaches to the myosin head. Through the enzyme ATPase, myosin ATPase, the phosphagen molecule is released from ATP to ATP, and the cross bridge then dissociates, allowing it ready for another cycle.
Where does it split? Like what part of the ATP goes where?
The phosphagen is used for energy. It's used, and the ATP is released, and it becomes rephosphorylated.
Can you explain again what is dissociation?
הפרדה. הילה זה הפרדה. להפריד בין mass and vacuum. Dissociation is a nice word, it's fancy.
What is the ACH? Your
Acetylcholine,
on a
neurotransmitter.
all right.
Okay, let's take a break here, and then we'll come back, finish up the lecture we talked about muscle types. 7 o'clock now, we'll go 11:15. All right? Sounds good. All right, guys, I'll put on some more music to listen to. I had, last time we used